Hi everyone, welcome. So today, me and my group will present about subtopic 5.2 nervous tissue, chapter 57 in medicine group. So I will introduce first my group members. So here we have Nazrumuddin bin Osman, AS11112. We have Adaru Aide bin Adharuddin, AS10753, and me, Ahmad Bati bin Muhammad, AS11235. So in our in, in our presentation, so these are the linear outcomes that we have to achieve in the end of our presentation. So we must be able to describe the basic structure of a neuron and compare the function of three types of neurons. Second, we will be able to discuss the change in ion concentration inside and outside a neuron that result in an action potential. Third, summarize the role of various neurotransmitters in propagating nerve impulse. So we have two types of physical types. We have two physical types of nervous cells. So the first one, we have neurons. Neurons we known as nerve cells, which is a functional unit of nervous system. So remember, first we receive the sensory information, and then we convey information to the brain, and then we conduct the signal from brain to effector structure. So here we can see the part of the structure of the neuron. Proceed to the second one, we have neuroglia. Neuroglia function as supporting cells, which providing support and the nourishment to the neuron. Okay, so neurons and neuroglia have three major parts of neuron. They have cell bodies that contains nucleus and a variety of organelles. They also have dendrites. They have dendrites to receive signal from the sensory receptors and transmit them to the cell body here. To the cell body from dendrites to the cell body here. Okay. So and then they have axon convey information to another neuron or cells. So as we know, they have bundle axons that called nerve fibers, and the axon covered by myelin shed. Myelin cells. This is myelin cell. The yellow one. Okay, so we proceed. So there are three main types of neuron. For the first one, we have motor neuron. Motor neuron is the afferent neuron that have multipolar shape, and they can transmit nerve impulse from CNS to muscles or glands. Second one, we have sensory neuron, which is we know that as afferent neuron. Afferent neuron is unipolar, and they will transmit impulse from sensory receptor to CNS. And the third one, interneuron. Interneuron is a multipolar neuron. They convey nerve impulse between various paires of the CNS from complex pathways in brain. So these are the other types of neuron. We have anasonic neuron here. We have bipolar neuron. We have pseudo-unipolar neuron and multipolar neuron. You can see the function. They have specific function between themselves. As for example, anasonic neurons have more than two processes, they have more than two processes, but the essence cannot be distinguished from template. For bipolar neuron, they have two processes separated by the cell body. The pseudo-unipolar neurons have a single elongate process with the cell body situated to the one side. The last one, multipolar neurons have more than two processes, and there is a single axon and multiple dendrites. And they have single axon and multiple dendrites. So I will proceed to the picture of nerve impulse with explanation of from Nazrumuddin. Thank you.
Assalamualaikum. Now I'm going to present about transmission of nerve impulse. So let's go. So before I'm going any further, let's know about this one. The picture that will be provided later. So this one is we know neuron. Then if we zoom into the axon, we will see axonal membrane. So there will be negative charge inside and positive charge outside. So if we zoom in again, and we can see inside axon and also the outside axon. So let's get started. So on this topic, we are going to know a few terms in this action potential. First thing first, we have to know resting potential. So what is resting potential? So resting potential is when axon is not conducting any impulse. When there is no impulses, the voltmeter shows that it will be negative 70 millivolt. So as you can see, these two wires, one indicate inside the axon, one is outside the axon. Outside. So there is preponderance of sodium ion outside the axon and a preponderance of potassium ion inside the axon. So as you can see, there is many potassium inside the axon and there is more sodium ion outside the axon. So because the membrane is more permeable to the potassium ions, which make it more positive ion outside the axon. Okay, because why is more positive outside the axon? Because membrane is more permeable to potassium ion. Now this one is resting potential, which is negative 70 millivolts. Now we go to the next action potential. So during an action potential, okay, what is action potential? Action potential is when depolarization, okay, this one depolarization occurs when sodium's gates open and sodium's ions begin to move inside. So as you can see, the sodium gates are open and sodium ions move inside. It move inside. Okay, it move inside. Okay, very clear. And as you can see, the potassium gates are closed. So that is why we call it as depolarization. So during this time, the axon potential will increase into negative 55. Before this is negative 17, right? So why cause this? Because of stimulus. Stimulus cause the axon to reach its threshold. So what is threshold? Threshold is minimum change. Okay, minimum change in polarity across axonal membrane that is required to generate an action potential, which means the stimulus generate an action potential. Because of the axon to reach its threshold. So this one is the beginning of action potential. So action potential start begins when depolarization it keep continuous until a potential of positive 35 millivolt is reached. Means the sodium will keep entering into the axon until it reaches to negative 35 millivolts. Because of the changes in polarity of sodium channel, because of the changes in polarity, it will cause the is the sodium channel to close and will make the potassium channel will be open. So that is the end of depolarization. Now we go to the next one, action potential, which is repolarization. Okay, remember repolarization occurs when, like I say, as you can see, both sodium gates are closed, but potassium gates are open. So it will make the potassium ion to move outside the axon. It move the outside. So as potassium leaves the axon, the membrane potential will down from negative 40 before. It will come back to the normal with its negative 70. Okay, that's why we of repolarization. The potassium ions move outside the axon. Now the last part of the action potential is refractory period. Refractory period. So what is this refractory period? Because as soon as action potential has moved on, okay, as soon as potential has moved on, the previous action undergoes refractory period. Okay, it will go under refractory period. So what is refractory period? Okay, refractory period is very important. It will make the sodium gates unable to open. So the action potential cannot move backward and instead will move down and axon to its terminal. Means it will make the action potential will move down until it reaches to the axon terminal. Okay. So that is the end of the action potential. Now we have to know about propagation of action potential. We have to know there is two kinds of axon, which are unmyelinated axon and myelinated axon. Unmyelinated axon, as you can see, there is no kind of myelin on the axon pathway. So during this time, as you know, the first thing that will happen is depolarization, right? Where the sodium enter the axon. So as you can see, it will enter the axon. It will keep continue. Okay, it will keep continue like this. It will keep continue. It will move down, and at this area will be repolarization. Okay, it will be repolarizing, which means the potassium ions move outside the axon. Okay, it will keep like this. Then it will keep move on. It will keep move on and it will go to the resting state. Okay, it will turn out to be like this. Why depolarized area will be repolarizing, and after this one, it will go to the resting state. Okay, so it will keep continue depolarize, repolarize and resting, depolarize, repolarize and resting. It will keep continue move down until it reach to the axon terminal. That's why is refractory period is very important because it prevents the action potential to move backward. Okay, there's no backward. Okay, it prevent action potential to move backward. Okay, no, and because of there is no masculine, this will consider as continuous conduction because there is no obstacle, and by it called as slow squeeze, which means it takes much more time, longer time to reach axon terminal. But in contrast, myelinated axon, as you can see this neuron, there is myelin on the axon pathway. So what is the function of myelin? The myelin is prevent current from leaving the axon. So when the current is prevent from leaving the axon, I mean it could increase the speed of action potential. So as you can see, this one is the area where the depolarized membrane. So because of the existence of myelin on the pathway, so it will jump. Pop, it will jump. Okay, it will jump to the next place on the resting state, on the resting potential. So it will keep continue to jump. It will jump. It will jump until it reach terminal as well. So because it keep jump, we call it as saltatory conduction, which means fast squeeze. Okay, it will reach the axon terminal in a shortened time. Okay, what is called as fast squeeze. It will jump over the nodes and on the myelin, over myelin. So that's why we know. Next we go to the we can to supply it. Can see it based on the graph. So this is graph based on the action potential. According to action potential, first at rest when the axon is not conducting any impulse. So at rest it will be negative 70 millivolts. But when the stimulus are applied, it reach to the threshold with minimum change, right? And it reach from negative 17 to negative 55. After it reach threshold, there will be depolarization will occur, where sodium ion will flow into the axon. So the voltage will rise until negative 30 millivolts. But when it reach positive 30, when there is changes in polarization of sodium, it will mean the sodium gas will close and the potassium gas will open. So the potassium ions will flow out into the outside of axon. So we call it as repolarization, and the voltage will fall. So it will keep continue until the refractory period. So during this time, there is end of the action potential. Okay, the end. So and so it will return to resting state. So this kind of condition will keep continue until it reach terminal axon. So transmission across the synapse. Okay, this part I will pass to the ideal to explain.
I will tell you about the transmission across a synapse. So let's start. So there are two types of neuron, which is presynaptic neuron and the postsynaptic neuron. The presynaptic neuron is the first neuron that receives the impulse, and the postsynaptic neuron is the second neuron. So between it have a gap called as the synaptic cleft. So the nerve's impulse cannot cross the synaptic cleft. So
what they can do is transmission across a synapse is carried out. By what? By molecules called neurotransmitter, which are stored in the synaptic cleft. Next, we move to the ways the transmission across the synapse. So first, the action potential arrives at axon terminal. Then second, voltage-gated calcium ion channels will open. Then the calcium enters the presynaptic neuron and the calcium ions signals to the neurotransmitter vesicle. Next, the vesicle moves to the membrane and dock. So this is the dock, the purple color, and this is the membrane of the presynaptic neuron. So next, neurotransmitters released via the exocytosis. And 7. The neurotransmitter binds to the receptor of postsynaptic neuron, postsynaptic cell. This is the receptor. And lastly, the signal initiated in postsynaptic cell. So next, the binding of neurotransmitter to receptor allows the sodium ion channel to open in the postsynaptic cell, allowing the sodium ion to diffuse from the presynaptic cleft into the postsynaptic bulb. If enough of sodium ions move into the postsynaptic bulb, then another action potential will be activated at axon. So next, the neurotransmitter. So there are many substances in neurotransmitter such as acetylcholine, norepinephrine, dopamine, serotonin, and endorphins. So acetylcholine excites skeletal muscle but inhibits cardiac muscle. Norepinephrine is important to dreaming, waking, and mood. Dopamine is involved in emotions, learning, and attention. Serotonin is involved in thermoregulation, sleeping, emotions, and perception. Lastly, the endorphins are neurotransmitters that bind to natural opioid receptors in the brain. It produced by the brain not only when there is physical stress, but also when emotional stress is present. Next, move to the next slide. In some synapses, the postsynaptic membrane contains enzymes that rapidly inactivate the neurotransmitter. The enzyme acetylcholinesterase breaks down the acetylcholine. In other synapses, the presynaptic cell is responsible for reuptake, a process in which it rapidly reabsorbs the neurotransmitter, possibly for repackaging in synaptic vesicle or for molecular breakdown. The short existence of neurotransmitter at a synapse prevents continuous stimulation or inhibition of postsynaptic membranes. Next, so the drugs affect the action of neurotransmitter because it can enhance or block the release of a neurotransmitter, mimic the action of a neurotransmitter, block the receptor, and interfere with the removal of a neurotransmitter from a synaptic cleft. Some antidepressant drugs, such as fluoxetine (Prozac), prevent the reuptake of serotonin. Bupropion hydrochloride prevent the reuptake of both serotonin and norepinephrine. Blocking reuptake prolongs the effects of these two neurotransmitters in networks of neurons in the membrane that are involved in the emotional states. So moving to the next slide. So this is the nerve. So this is an axon hillock, the area where incoming signals are integrated. So the integration is the summing up of the excitatory and inhibitory signals. If a neuron receives many excitatory signals either from different synapses or at a rapid rate from one synapse, chances are the axon will transmit a nerve impulse. So this is the end of our presentation. So thank you for watching and listening to us. See you next time. Bye.